record ko. Okay, hi, good afternoon naman noon. So, tay sa kadali mag-ano sa day ko. Mag
Si Brian.
mag-attendance. So, welcome to your subject in that side 22. We are now on our day 5 in our lesson. So, your prelim exam is coming up. So, please, if you can settle your account soon, you can settle, then I can send you your prelim test. So, but we still have the whole semester anyway, so if you cannot do it now, so you can do it later whenever you're available, no? So we are actually done with the scientific method from our previous topics. So I already uploaded a video, so you can check this and review the lessons that our presentation that we have. So for the next lesson, we're going to talk about the microscope, as a whole, and the uses of the microscope. So before we start, do you have any concerns regarding the quizzes, Jerome?
Okay raman ma'am ang katong quiz. Wala mo kay question nato.
Okay. So actually, I have not checked yet your answers, but hopefully this coming weeks. So but then you will always be updated with your score anyway online, and if possible, this attendance would be also your additional points to all of your grading system, because your participation during meeting is like for discussion or interaction would also count, although it's not really included but additional points. So of course, you know what is a microscope, diba? For you,
Yes, ma'am.
Jerome, what is the importance of microscope in your life, in your as an agriculture student?
Mm, the importance of microscope, for example, if in the future I will be like in the research, so I can make use of this instrument to like look the microorganism that can't be seen by our naked eyes. So we'll have to use the help of the microscope to much see what's in the like the microorganisms that you're going to do the research on.
Very good, very well
Or
said. Yes, some more?
kana lang.
Okay, so anyway, although you are not directly involved in the research process, or right now we're not having experiment on this, that's why on your not sci laboratory exercise, I hope you have seen already the lab guide. The lab exercise one, instead of using the microscope, is to look online videos or visit a laboratory near to you. If possible, you are accessible to any laboratory. A biotechnology lab, example of biotechnology lab is a genetic lab, a tissue culture, no, or they call it biotech lab, no, wherein you can find a lot of equipment, not only microscope. So if not, you're not accessible, like you're in Agusan, no? Agusan ba to?
Surigao, ma'am.
Ay, Surigao pala. So you're in Surigao, so you can just look online videos, and you just Google, search on YouTube or Google biotech lab, and in that way you can learn the equipment, and what they are doing in the biotech lab. And there are series of guide questions in your laboratory exercise that you're going to answer and report. So anyway, as a review, because in your maybe on your high school or primary education, you have learned already the parts of the microscope. So the importance of microscope before we proceed is um is related to our past history, because we will never know what cell look like and what genes look like without the discovery of the microscope, and we cannot move forward to genetic you know because we are now on biotechnology, genetic engineering, and we cannot move further on that without knowing or understanding the microscope. That's why understanding and knowing the function of the microscope is a key to understanding life, no, or biological living things. So of course, this is the eyepiece wherein you can see. Of course, have you tried using the microscope before, Jerome?
Yes, ma'am.
Okay,
Yes.
so maybe you have seen already what whenever you look into the eyepiece, so it is where the specimen or the cell specimen is clipped here in the stage clip. So this is the diaphragm or the stage, and it is with the this mirror must be faced on the light side or whenever there's light or sunlight so that you can see what is reflected here. So these are the objectives are the different, you can turn around the nose piece, so you can change the objectives into how many times, how many times, 10 times or 5 times and more higher resolution, so you can see um more clearer or um uh more what they call that detail or near or far distant view of a particular object. So this fine focus will also help you adjust this knob here, to make the objectives clearer or finer, and the coarse objective to make it farther. No, so these are just the important parts of a microscope. We have more high-tech microscope like the electron microscope, which will worth billions. No, and I think the only available electron microscope here in the Philippines is located in UPLB. I don't know if it's in Erie or in their biotech or in genetic engineering lab. So microscope is used to produce an enlarged image of an object. So it's an equipment actually to study organisms, cells, and cell parts, and just to increase their size. We call them magnification. And the ability to show details clearer is called resolution. Yung mga nose piece, those are the resolution. So when you can just define microscope or microscopy simply as to make an image, or to produce an enlarged image of an object. So as I have said before, microscope can be so high-tech, so they vary in both magnification and resolution. So yung mga electron microscope are actually very high resolution na. So this one I have presented to you before, and this is to show you the nanometer um, number of cells, found in all living organisms, from very small to the biggest number, up to 1 kilometer. As I have told you, humans have is second in terms of um, size of the cells. So there are four types of microscope, and the only one that we have is a simple. And I have not checked actually here in the lab. I should have shown you. So next time I'm going to. I just forgot to take off the microscope so I can show you here in this video. But we're going to do that later. The simple, the compound, and stereoscopic and electron microscope, and we're going to discuss them one by one, each differences. So the simple microscope is just similar to magnifying glass and has only one lens, no, like this. So have you seen a magnifying glass? Have you seen a magnifying glass?
Yes, ma'am.
Yeah, so
Yes, ma'am.
it's just similar to that. When you just use a magnifying glass, then it's just simply make the object larger. And we have the compound microscope. I think this is what we have here in our college, no? It lets light pass through an object and then through the two or more lenses. Ito yung mga lens, no? Ayan. Another one is, yeah, this we also call this compound light microscope, wherein specimen is mounted on a glass slide. So I I believe you have seen already a glass slide. It's where you put the cell or the tissue and then you cover it with a cover slide, which is very thinly sliced or small. And then in the, when you clip that here on your skin on the stage clip, and through your lenses, your pair of lenses are the ocular lens, this one, and the objective lens. No, these are the objective lenses. So you have two lenses actually. The ocular, the one that you put in your um, eyes, and the objective lens here or the nose piece. And these are used to magnify the specimen you are studying. So this magnification determined by multiplying power of both lenses. So when you report, like when you have seen this cell and you use the 10 times eyepiece, so you report them at 10 times, no? 10 times objective power or 20 times or 40 times. So highest magnification is around 1,000 times. So wala, I don't think we have seen, and me myself, I haven't seen only in photos, no, this 1,000 times because I haven't used an electron microscope yet. Aside that it's a limited and it's expensive, no, it's only this is the electron microscope. Have you seen this before in any photos or
No.
video? No. So it's
Electron
so,
manus wala.
yeah, it's so huge, and it uses a magnetic field to bend beams, diba, in a simple or a compound microscope. Um, this one, this light source here, this will condense the light here and bend so you can see the specimen. But in the electron microscope, you use magnetic field to bend beams of electrons instead of using lenses to bend beams of light. So it's so huge, no? It's even bigger
Yes.
than a human person. So the electron microscope has transmission or EM. Transmission EM. Um, Produce an enlarged image of a very thinly sliced specimen on screen or photographic plate, and the image focused by magnetic lenses. So instead of a natural glass lens, you use magnetic lenses. And it's 200,000 times magnified, no, or magnification. Imagine 200 times, 200,000 pala times. Yung compound microscope, the highest lang resolution is 1,000 times. But this electron microscope is 200,000 times. but cannot be used to view living specimen. No, unlike the compound microscope, you can see them, no, swimming, like the amoeba, you, the single cell bacteria, and so on. You can see them swimming on your slides of glass. But in electron microscope, they are not for living specimen. So this scanning EM is a three-dimensional image. Have you seen a three-dimensional? So it's like this, no? Parang when you make your photo a 3D image, no? Facebook has an app for that. Have you tried that one in your Facebook?
Murag
Yes.
360 ma'am. Murag
Yeah,
isa to.
you can, yeah, that's a 3D image, no? You can see the sides of it. Specimens not sliced for viewing. Surface spread with fine metal coating, and also uses electron beam and fluorescent screen or photographic plates. And it's 100 times magnification. So actually, this had been mentioned earlier. So stereoscopic microscope, this is another type of microscope with, which gives a 3D or three-dimensional view of an object also. And these are used for insects and leaves. So this one, celestron, no? It's just so cute, but it uses a lens, enlarges an image, and bends the light toward your eye. Para siyang para siyang this one, no? Yung eyeglasses mo. So this is just to show you how the light is bent using these lenses. So the eyepiece lens, this one, or the eyepiece lens, or the resolution usually has a power of 10 times only. And this eyepiece lens is your objective lens. So if it's 10, so you just put X, that's your total magnification, meaning 10 times, so 10 times the size of that object, or 20 times the object. So these are the example of objective lenses. When you say low power, if you remember, we have three in your compound microscope, you have three lenses, no resolution or nose piece, which is classified as low, medium, and high power. So yung low power is 4 times, medium is 10 times, and the high power is 40 times. So in so those are just the microscope parts, no, and types. Now we before we proceed with the next one, which is the microscope microscope specimen. Do you have any question,
Wala man ma'am.
Jerome? So, ang wala ngayon is si Beam at saka si John Ray, no? So, actually, we moved the schedules for John Ray kasi John Ray is working. So, that's why we're having a lunch break,
Si Brian.
working break. But, nag-lunch na ka?
Wala pa ma'am.
So, kuan sa ta karon, mag-fasting. But I'm done with my
Okay
lunch
lang ma'am.
already. So the study of microbial structure, microscopy, and specimen preparation. So this will show you again, this is a nicer um, view of how, of the sizes of the different cell structure of organism, no? As I have mentioned earlier on the previous slides, from zero point 0.1 nanometer, 1 nanometer, 10, 100, micro, 10, 100, zero point 0.1, no? So this organism, using unaided eye, uh, you can see this, the human height, length of some nerve and muscle cells in the chicken. So that's uh, and up to this one, from 1 millimeter frog egg, you need a light microscope for that. no, up to this mycoplasma, eukaryotic cells, nucleus, most bacteria, and mitochondria. And you can only see this under light microscope, no. So 1 m to 1 millimeter, you can still see that using your eyes, but lower than that, you need a light microscope. And from this 100 microns, you need an aid with an electron microscope to see them, no? These viruses, mycoplasma, of course, this one is included, but further down, 100 nanometer is mycoplasma, viruses, ribosome, proteins, lipids, small molecules, and atoms. It's all visible under electron microscope only. So it was on 1632 to 1723, when the first person to observe and describe microorganism accurately, by Anton van Leeuwenhoek. So this is a very familiar name, has been mentioned many times in our biology. Said that it was Anthony Leeuwenhoek who discovered microorganism using the microscope. So, light is refraction ang tawag dyan, or bending, when passed from one medium to another. So that's how lenses and bending of light is done by the lenses of our microscope. So refractive index is the measure of how greatly a substance slows the velocity of light. We know that the fastest speed is light, right?
Yes, ma'am.
Mm. So the direction and magnitude of bending is determined by the refractive index of the two media forming the interface. So um, that's why there's an index in the refraction, no, because of that's the magnitude of bending. So if you notice, even the lightning, no, before yung thunder will you can hear the thunder after, or even minutes after the lightning, because the lightning or the light will pass. It's the fastest speed on earth, no. It's so maiwit ang thunder kumpara sa lightning.
Under.
So lenses, the focus light rays a specific place called the focal point. So if you notice, meron siyang bent ang lens, and it's called focal point. It's the distance between center of lens and the focal point is the focal length. The strength of lens related to focal length is the shorter the focal length, the more is the magnification. No? So if the focal length is longer, yung magnification naman niya is lesser. So it's about the lenses, this one. The curve here. So if the curve is shorter, like ana lang ang curve, meaning the magnification is higher. Pag curve kay siya. Pag longer yung curve is higher, lower yung magnification. So there are different types of light microscope. Could be bright field, dark field, phase contrast, or fluorescence. And this is just to show you, no, because you will never pass this again, no. If you go deeper, so although these are actually very comprehensive study of the light microscope. So, but then you should be able to know, marami pala yung types. Our compound microscope, image formed by action of greater than or less than 2 lenses. So for compound microscope, it's about more than 2 lenses. That's why it has um, yung three nose piece kanina. So the bright field microscope, it produces a dark image against a brighter background and has several objective lens. called parfocal microscope remain in the focus when objectives are changed. And total magnification is the product of the magnification of the ocular lens and the objective lens. So once again, yung nakikita mo sa microscope are the products on your nose piece and your ocular lens, no? So for a bright field microscope, it's just a dark image against a bright background. So this is an example. So we have of the compound microscope, which is a bright field microscope. Yan. So as you can see, it has more comprehensive parts too, no? And it even has 4 objective lens. Apat siya.
Duha pod ka kuan kanang eye sa naman
Yes. Double ocular eyepiece.
eyepiece.
Yeah. So, in a normal, kung wala pa sanang pandemic, so we will have activity sana on how to use the microscope using different single cell bacteria under the microscope. So you would be able to see, and even have you tried yung hair strand under the microscope?
Mala, ma'am.
Kasi it's a very simple activity done in the high school, no? In biology class in high school. So, talking about microscope resolution, it's the ability of a lens to separate or distinguish small objects that are close together. And wavelength of light is used as major factor in resolution. The shorter the wavelength is, the greater the resolution. So, are you familiar of a wavelength? Are you familiar?
Wavelength, ma'am.
Yes.
Familiar lang siya sa kuha, light used in a major factor.
So we have different wavelengths, no? Yeah, these are the wavelengths. I have here an example. This is my favorite photo actually of a wavelength. So this is the wavelength. Music, sound, gamma rays, and ako na lang siyang save. Gamma ray, gamma rays, and this one, microwave, radio waves are all wavelengths. So as you can see, ang light na to, let's start with the most destructive wavelength, no? This is the 10 to the 6th nanometer, so the gamma rays. So if you have heard about the, what do you call that, a radioactive birth before, katong radiation na siya, no? So it can kill a lot of person, no? Or yeah, like in the what happened in Chernobyl, no? In Chernobyl, if you have heard that before, if not, then it's somewhere. I think it's somewhere in Russia. I'm not so sure. But I'm going to, or you can also look on that, no? There's a radioactive nga nagbuto, no? So with the gamma, it's made of radioactive gamma rays that destroys, parang mag-malform na yung mga tao. There's a lot of movies on that, no? To na if you have
Wrong
seen,
turn, ma'am.
yeah, if you have seen movies nga
Wrong turn.
mura na silag mga alien, it's because
Mutated
of the,
na, nag-mutate na.
yeah, it's destructed na. And x-ray, kanang x-ray na ginagamit mo nato if we're going to the hospital and scan our body structure, our bone, no? So those are the x-rays. And these are destructive already. In even the UV radiation, the ultraviolet radiation. That's why we use UV for disinfection, to kill bacteria and other organisms, even insect, pests, and viruses. So from here up, it's very destructive. And this is the, what we call as the light rays, no, wherein these are beneficial to human living things or human, no. So this is also the visible light where we can see on the from the sunlight. So it's in accordingly from 400 nanometer to 700 nanometer. So each rays can be color have color like red, orange, yellow, green, blue, violet, no. That's why yung UV malapit siya sa violet, kaya purple ang UV light. So we also have infrared radiation, microwaves, and radio waves. These are less ano na siya. So these are all wavelengths of light. Ha? So if you can notice, one of the wavelengths of light is the radio waves, where we hear frequency and kanagaling sa radio kanang madungog na.
Oo, kanang magkuhan ka mag
Yes, because
mm.
they have very high wavelengths. That's why we can hear them. So that's what they use in AM, FM radio. Mo na siya. Kaning microwave also are what we use when we heat the food. And there were actually debates on. They said microwaves are dangerous, no, or risky to your health. But um, there's no studies yet to prove that exactly, but we never know yet, no, and until now. But by uh, by theory, the microwave is not actually as that destructive. Uh, so going back to the presentation, so that's the wavelength. I don't know if someone's calling this at decline sa nako. So that's about, so the shorter the wavelength, the greater resolution too. So if you remember, the shorter the wavelength, these are the short wavelengths. It's also destructive, di ba? Wait lang ha.
Mm.
Inan sa nako nila nag-classic. Okay, so next is the properties of microscope objectives. So these are the properties: the magnification, the numerical aperture, the approximate focal length. So we have discussed about focal length kanina. It's the curve of the lenses, the working distance, and the approximate resolving power with light. No, under 450 nanometer, or this is under blue light. So, and its specific objectives: scanning, low power, high power, and oil immersion. So these are just the characteristics. If it's scanning, the magnification is 4 times. Pag low power, 10 times. High power, 40 to 50 times. 90 to 100 times pag oil immersion, and so on. So working distance is the distance between the front surface of lens and surface of the cover glass or specimen. But then the cover glass is just covers the specimen. Depende yan sa how thin is your cover glass. So this is the objective, the working distance, like from your resolution and the slide specimen. So what is the working distance from here to here? So usually the working distance from 17 to 20 mm, 4 to 8, and so on. So this will also show you again the distance for the oil immersion, no, from the distance from the cover glass, and it has always, although when we close them, you can see that it has no space for us already, no, from the glass line, but to the cover glass. But then you should always put some extra space for air, because air has space also. Even when you close under a closed bottle, that's why lahat ng food packaging meron siyang what they call that, na siya sa tawag na siya, oy kanang uh, lifespan, no? Kasi later on, even bottle siya, but when you close a bottle, but you may notice na after how so long or how many years, madaot gyapon ang sulod sa bottle, di ba? Although closed pack
Mm.
siya. But it is because through the structure ng glass bottle is made up of different different structure or matter na pwedeng there are organisms that can still penetrate through the glass, no? So it could be, although airtight na siya, no? So those are uh the light field microscope. We also have the dark field microscope, which produces a bright image of the object against a dark background. So it's the opposite of the light field, no? This one, the bright field pala microscope. So the dark field is just the opposite. Kanina, it's the contrast between kanina, it's a dark image against bright image, but this dark field is produces a bright image of the object against a dark background. And this is used to observe living and stain preparation. So this is just to show you how is the dark field, no? Um, objective and the lenses and the specimen and the condenser is to show you, no, in illustration. So in illustration, this shows that the bright image, this one under this is under against a dark image. So another type is the phase contrast microscope. It enhances the contrast between intracellular structure having slight differences in refractive index, and these are very excellent to observe living things because there's a contrast between intracellular structure, and it just have slight differences in refractive index. When you say refractive index, yun yung kanina, the total magnification index. So this will show you the dark image with bright background results, no? Yung how the light is bent, no? And how it goes through your ocular lenses. So this is another, like for bacteria, how bacteria is being, if this is your face plate, the ray here
Face
is
plate.
deviated by specimen like 1/4 wavelength out phase. So ayan. And then its deviated ray is 1/2 na wavelength. And then that's how you see the undeviated already rays. So another one is differential interference contrast microscope. It creates image by detecting differences in the refractive index and thickness of different parts of the specimen. And this is also another excellent way to observe living things because you use differences in refractive index and thickness of the specimen. So you use the fluorescence microscope. This exposes specimen to UV, violet, or blue light, and specimen usually stained with fluorochromes, and shows a bright image of the object resulting the fluorescent light emitted by the specimen. So if you have, are you still using fluorescent light or LED light na sa inyong bahay?
Naapay fluorescent ma'am gihapon diri sa balay.
So for you, what is the difference ng in terms of sa lighting, fluorescent light at saka LED light, okay, when you still have fluorescent light there?
Ang fluorescent ma'am kay kuan kanang dili siya kanang instant gud damuon diretso. Dili
Kasi
pareha sa
meron siyang
LED.
balas,
Pagman
'di ba?
na pa siya balas, na pa siya starter.
So for you,
As well as kung LED na.
Mm. But for you, which is lighter? I mean brighter, the fluorescent light or the ano? Although meron siyang different ano no, different characteristic at saka
Watts
yes, explanation
ang
behind.
wattage.
Mm-mm.
Mm.
So, but then for now, we use LED light because they say, science say that LED lights are more efficient and can conserve energy. No, and it's also
Mm.
environmental friendly compared to fluorescent light. So, fluorescent light also
Mm.
emits different wavelengths, no? As you can see, all the lights, meron yung wavelengths from 400 to 700 nanometer from the
Mm.
yellow, red, blue, green, at saka purple. So this is just to show you another figure on how this from how the light is bent no through the mirror. No? So mubaliktad yun na siya. So if you can see kanang makita sa nimo sa lens no or even in your eyeglasses or even in your camera sometimes, i-bend mo na sa mirror, and once it's bent, mabaliktad yun na. Ang makita nimo is the is the opposite of the image. So this is just to show you the figure under the microscope, no? Yan yung makikita mo sa microscope. This is what you can see. So how do you prepare the specimen, no? Which will increase visibility, accentuate specific morphological features, and preserve specimens. And we call them fixation, by which internal and external structure are preserved and fixed in position. So it also has a skill that when you try to cover your glass slide, I mean your cover to your glass light, it must be fixed enough, no? Not to alter the structures internal and externally. It's also a process by which organism is killed and firmly attached to the microscope slide. So sometimes there are already available commercial glass slides, no, like eukaryote cell, prokaryote cell, pwede na siya commercially available, order lang nimo para makakita nimo kung unsa ang cell sa eukaryote and prokaryotes. And it is done through heat fixing, preserves which preserves overall morphology but not internal structure, or chemical fixing protects fine cellular substructure and morphology of larger, more delicate organism. That's why in bacteria, para you can identify bacteria of two types, whether meron kang i-add na yung purple, I forgot, blue thing. So pag i-color nimo ang bacteria, then you will know if it's this kind of bacteria or not. So that's called chemical fixing. Yeah, this is the one I am talking about. When you stain them using dyes, make internal and external structure of the cell more visible by increasing contrast with background. And it could be chromophore groups, chemical groups with conjugated double bonds, which give dye its color and bind cells, or simple staining using single staining agent like basic dyes, like crystal violet, frequently used dyes with positive charges. So they can be like for bacteria, if you use dye for them, they can be gram positive or negative. So there are two groups of bacteria, whether gram positive or negative, and divides microorganisms into groups based on their staining properties, or acid fast stain, no? Hindi lang gram stain. So what is gram staining? It's the mostly widely used differential staining procedure, which divides bacteria whether positive or negative based on differences in cell wall and structure. So this is an example. In your plant, are you done with plant pathology subject?
Yes, ma'am.
So, have you tried to stain bacteria before under the microscope?
Wala. Wala may kagamit o microscope
So,
atong
anyway, if you can isolate the bacteria and you put that on your slides under the microscope and you put dye, like for crystal violet, for example, for 30 seconds, then water rinse for 2 seconds, this is a primary stain. And grams iodine for 1 minute, then water rinse, this is more than. Then wash with 95% ethanol or acetone for 10 to 30 seconds. Then water rinse to decolorize. Then safranin for 30 to 60 seconds. Water rinse and
counter
blunt
stain.
as counter stain. So if they color purple, then they are positive. If not, then they are negative. They belong to the gram-negative bacteria or positive gram-positive bacteria. For example, the Escherichia coli. Are you familiar with Escherichia coli or coli, whatever?
Nope.
So it is a bacteria
Dili,
that
ma'am.
is no yeah, Escherichia coli is a bacteria that is mostly found damaging plants or in humans as well, no? It's a gram negative rod. So it's gram negative. So it doesn't color purple under the microscope. So medyo pinkish siya. So another, aside from gram staining, is the acid fast staining. This is for staining members of the genus Mycobacterium, like the Mycobacterium tuberculosis, which causes the tuberculosis, and the Mycobacterium leprae, which causes leprosy. So it has high lipid content in cell walls responsible for the staining characteristic. So how do you stain using this? Uh, if it's negative staining, this is often used to visualize. This is for staining specific structure. Negative staining is used to visualize capsules surrounding bacteria, and capsules are colorless against a stained background. If it's spore staining, it's a double staining technique. Bacteria and the spore is one color and vegetative cell is a different color. And or flagella staining, more than applied to increase thickness of flagella. So that's for the staining procedure. Now, let's just let's just wait for this video to stop, no? So if you have questions about electron microscope, I mean, types of microscope, um, specimen preparations, before we proceed with electron microscope, because this is another topic that we're going to discuss, no, on, because we cannot finish this one for now. So, may
Mm.
tanong ka ba about staining?
Kanang staining karon pay ko kadungan nga mga naday ng staining
Well,
about
at
like
yeah,
kanang mag
yung
kanang
gram staining for bacteria.
mm,
It's a basic knowledge in biology, no? But this, because this subject is agricultural biology, so it's more applied science. So anyway, at least you
precision
have
ko.
known, at least even if you don't remember gram staining, later on, like ano ang kaibahan ng gram staining sa acid fast staining. So at least you have remember that you have come across with about may staining pala in
Staining.
for bacteria.
Mm.
And dalawa lang naman yung types, no? Yung gram kanina, yung gram, whether gram staining, whether gram negative or positive siya. And for acid fast staining, it's only used for mycobacterium, which is very important in medical technology, kasi mycobacterium na yung nakakos ng tuberculosis, yun yung ginagamit nila to identify this mycobacterium tuberculosis. And even the leprosy, no? So,
Mm.
wala ka ng tanong?
Wala na, ma'am.
So, I guess we will stop muna for the meantime. Or do you want to proceed?
Sa next na lang siguro na una, ma'am. Para
Lagi ka,
ang
ikaw ra isa na.
katong wala. Masig makakuan.
Yeah, so we will proceed
Mm.
with electron microscopy next meeting because it's another topic also kasi kanina from we have done a lot of discuss a lot of topics about microscope, no, kung when you when you speak of microscope you just say, oh, it's just a microscope is just an equipment, no, but now
Mhm.
there's a lot of,
Depende kaayo.
yes, no, it's not only the
Complicated
8
kaayo.
parts or 10 parts of microscope. When you say parts of the microscope, you just say the lenses, the resolution, the arm, the the stage, no,
Mm.
and the base.
Ito.
Parang anim lang yan, but no, it's not only 6 parts. There's a lot of it. So, thank you for listening, Jerome. And kung wala ka na
Mm.
talagang tanong, then we can end. Thank you. And I hope to see you
Thank,
next meeting. Wala na dyan kay
yes, ma'am.
pangutana.
Wala na, ma'am.
Okay. So, I also still have class next after this. So, see you again next meeting.
Yes, ma'am. Thank
Bye.
you, ma'am. Bye. Thank you.